Alright guys, in this video we're going to finish up our protist and fungi notes, and this will be the end of the notes for this particular unit. You're going to be testing next week. So we've already covered protist, and once again protists are kind of like a misfit group. They used to be a kingdom, but they aren't a kingdom anymore. They're very diverse. The only thing that all protists, have in common is that they are eukaryotes. So we've covered our prokaryotic kingdoms, bacteria and archaea, and then now we have started covering our eukaryotic kingdoms. So protista were mostly unicellular, mostly aquatic or in moist environments, but the only thing that they all shared was that they are eukaryotes, which means they have the nucleus and they have the membrane-bound organelles. Now, as we continue on, everything else that we're going to talk about is also going to be in domain eukarya, kingdom eukarya, as are the fungi, okay? So one of the things that we talked about with protists is that they were either plant-like, animal-like, or fungus-like, and most of that had to do with their nutrition. Now, fungi are definitely more of a cohesive group. They definitely have some characteristics that put them in this group. And so they are, of course, going to be eukaryotes, which means they have the nuclei and the organelles like mitochondria. And they do not have chloroplast, though. We'll see that here in a minute. Now, they are mostly multicellular. So fungi are kind of the opposite of protists. Protists are mostly unicellular. Fungi are mostly multicellular. But there are some exceptions. Your yeast are actually unicellular fungi. This right here is a very important defining characteristic. So I mentioned back with prokaryotes that the cell wall is really important in classification. Your bacteria had peptidoglycan. Your archaeans did not have peptidoglycan. When we looked at protists, we really didn't talk about cell walls too much because it really depends upon the protist type that it is. If it's plant-like, they probably have cell walls. If it's fungus-like, they probably have cell walls. But if it's animal-like, they won't. So as we talk about fungi, this right here, this word, is called chitin. I know it doesn't look like that, but chitin is the main component in the cell wall of fungi. This is the same material that you'll find in insects in that crunchy exoskeleton. So it is a carbohydrate. This is not typical, but it's a carbohydrate that contains nitrogen. Normally carbs don't have that substance, but chitin is definitely a word you need to always associate with fungi. Now, they are heterotrophs, which is why they don't have those chloroplasts. They do have mitochondria, but they do not have chloroplast because they cannot make their own food. And this is another major feature of fungi, is that they obtain nutrients by absorption. Now, in order to be heterotrophic, that just simply means that you're not making your own food. And so where a consumer would eat, like physically consume and chew and break down and eat, your fungi are a little bit different. Okay, so they're not making their own food, but they're not necessarily consuming like an animal would. They are actually absorbing their nutrients. Okay, this is why we call them saprobes. Saprobes are going to absorb. So what they do is they secrete enzymes. Hydrolytic just means they're breaking materials down, okay? That comes from that word hydrolysis, where you add a water molecule to break. But essentially, the fungi secrete enzymes into their surroundings. They kind of break down those materials, and then they absorb the nutrients that they need. Now, a lot of times these are decomposers. Typically, we associate fungi with decomposition, but they could also be parasitic, and they could break down tissues on another organism. Or there's also a lot of good fungi out there, and we're going to take a look at some of these ecological roles. So when it comes to the structure of a fungus, this is definitely different. When we talk about cells, a lot of times we will look at a typical plant cell or a typical animal cell. But with fungi, it's not that they don't have cells. They are cellular. All living things are cellular. But their structure is a little bit different than what we're familiar with, okay? There are some components that look a little plant-like, but they are not the same as like plant cells. So what they have are these little tiny thin filaments known as hyphae. Okay, and the hyphae will make up the multiple components of the fungus. So typically you'll find a mass of hyphae under the ground known as the mycelium. Okay, and the mycelium is where they're going to get a lot of their absorption. Okay, these hyphae are going to be very absorptive, and so this mycelium is just a huge mass. Okay. By the way, I want to go ahead and point out that this prefix here, myc, M-Y-C, we're going to see that throughout the fungus unit. You have myc, myco several times. So the MYC is often in reference to a fungus. Okay. So what the mycelium does is, because it's this huge mass of hyphae, it actually increases surface area. It's kind of the same concept as a root system for a plant. The more fibrous the roots, the more water they can absorb. It's the same kind of concept, but it's not the same structure as a root. Now, these hyphae are these long, thin filaments. Typically, they're divided into sections by cross walls, and we call those septa. However, not all of your fungi have these septa, okay? We call them cenocytic fungi, and that they don't have these cross walls. Instead, it's just one long continuous mass of cytoplasm on the inside of these hyphae. So under the ground, you will typically find the mycelium. Above the ground is what we call the fruiting body, and this is normally the reproductive structure. The fruiting body is normally what produces the spores for reproductive purposes. Now, if you think about probably the most common fungi, you're probably picturing a mushroom. And so that mushroom that you see growing out in your front yard or backyard or in the woods, that's the fruiting body, okay? And then some of your fungi have what are called rhizoids, and it's kind of a modified type of hyphae. The prefix rhiz, R-H-I-Z, normally refers to root. So these are not true roots, but they're kind of root-like structures that can help anchor the fungi into the soil or into like a tree or something like that. So here's an example of what they look like. So like I mentioned, they definitely are kind of root-like. If you were to look at these, you would probably mistake them for plant roots. But these are just masses of these little tiny thin filaments. So this picture up here, this is a microscopic image, and so you can see they must be really, really tiny for them to look like this under the microscope. And so under the ground, we have this mass of hyphae called the mycelium, but these hyphae also make up that fruiting body, which you'll find above the ground for reproduction. Now, when it comes to fungal reproduction, they reproduce with little cells called spores, okay? Now, these spores can be produced in different ways. They could produce them sexually or asexually, but spores are everywhere, okay? This is something that it's kind of like bacteria. They're in the air, they're on surfaces, and so just to kind of give you an idea, you're probably familiar with mold growing. And if anytime you have like dark, damp places, a lot of times you can have molds and mildews growing. And that's because those spores are everywhere. Those spores are actually on you, they're inside of you, they're in your mouth. These fungal spores are microscopic, and they can spread far and wide. What they're doing is they're just waiting for the right growing conditions. These spores can remain dormant for long periods of time. So if you think about like bread in your pantry, I mean, that bread can be wrapped up in a bag, sealed up in a pantry, and yet mold can still grow on it, because anytime it's exposed to the air, there could be fungal spores that land on it, and they're just waiting for the right growing conditions. So they spread easily over great distances, and they just look for the right conditions to kind of germinate and grow into a new fungus. Now, I'll be the first to tell you I am not an expert on fungal reproduction, so I couldn't tell you all the ins and outs of all of these. These are just some basics. So all of these here that are listed are asexual means of producing spores. And so they can produce spores in these structures called sporangia. They could form these spores at the tips of hyphae. But remember that if it's asexual, they are genetically the same, okay? They could bud. So down here you see pictures of yeast budding, and that's where essentially they kind of pinch off another cell that's identical. or just simply a piece of that mycelium, that underground hyphae, could break off and it could essentially form a new fungus. Now, when it comes to sexual reproduction, sexual always involves genetic information from two different places. Now, it's a little bit different with fungi in that they do not have male and female. They have different mating types, which, if you look at the pictures, a lot of times they represent that with a plus and minus. So they don't have like sexes in terms of male and female. They just have different mating types, which just means their genetics are different, their nuclei are different. And so, in terms of sexual reproduction, we just have to have a combination of genetic info from two different places. So, you could have hyphae of two different mating types that they could kind of grow towards each other and they could fuse. And so, if hyphae from two different fungi fuse, that leads to genetically different nuclei. And so, this word here, heterokaryon, hetero means different, karyo means nucleus. And so if you have hyphae from two different fungi fusing, we now have more than one type of nuclei in those hyphae, and that's where you get that term heterokaryon. Now, a lot of times this involves plasmogamy, the fusion of cytoplasm, as well as karyogamy, which is the fusion of nuclei. So back when we covered meiosis, we looked at some different life cycles, and the fungi is definitely different from what our life cycle looks like. And a lot of times your fungal groups can switch between sexual and asexual. It just kind of depends on the conditions at the time. So if it's asexual, they're just producing spores, pretty much keeping it the same. A lot of these would be like mitotic divisions. You could have meiotic divisions. You could have fusion of nuclei. You could have mating types coming together. But the key to sexual reproduction is you are going to have meiotic divisions. Whereas over here, it's just going to be basic mitosis or processes like budding. Now there's 6 major groups of fungi, and we're going to take a look at some of these in your lab activity. Some of these are maybe considered their own kind of phylum. You know, we're all in kingdom fungi. And it's kind of like with the protist. You're going to look at this more closely in lab and look at some examples. We're just going to take a look at some of the general characteristics. So the first group are microsporidia. Okay, so that literally means little spores, but these are unicellular. Okay, that's why they're micro. They're tiny fungi. There's some parasites that live inside of animals. Your chytrids, they actually are flagellated. These are an aquatic type of fungi. They have flagellated spores and gametes, and so once again you could see some parasitic chytrids, or you could see some absorbers. That's what a saprobe is. Your zygospore. Okay, now zygospores, most of your fungal groups are grouped based on their reproduction and the structures that they produce. And so your zygospore fungi produce these structures called zygosporangia, and they're actually very resistant. Okay, so these are going to be like your common molds. If we were to put this in a kingdom name, these are called zygomycota. But a lot of your molds are going to fit in this category. Some of them are parasitic, some of them are good. They could be mutualist. But essentially they're grouped because of this resistant structure. And so like I told you, these mold spores are just kind of hanging out waiting to grow. And so they can remain inactive for long periods of time. And then once the conditions are right, they can kind of exit that dormancy and actually begin to grow into the adult fungi. So as I said earlier, I'm not a fungal reproduction expert. All I really want to point out here is this structure. So this is what puts these fungi into this group, is they have these resistant zygospores, or these zygosporangia, and these are the structures that are going to be their spore producers. And so that's what puts all of these organisms into this group. Your next group, Ascomycota, once again, you have see that MYC, mycota equals fungi, or MYC equals fungi. And the way I remember this one is if you take the first 3 letters and you rearrange them, you get the word sac. And that's because they make sac-like structures called asci. Okay, or ascospores. They are ascocarps. And so once again, it's all about that reproductive structure that they produce. So yeast, some of your yeast fit in this category. Cup fungi that you might see growing outside. Now morels and truffles are both edible fungi. This is a morel right here. It kind of looks like a brain. They're kind of like your more decadent type of mushrooms. You're not going to find these at your basic grocery stores, but you'll find these kind of in fancier type cooking. Some of them could be pathogens, some of them could be lichens, so you can see there's a wide variety. But once again, the one thing that holds them together are these ascospores and that are produced in these ascocarps. And so here's a look. This is an example of a cup fungi there, but their reproductive structures are different. So here you can see your little ascospores that are being produced inside of the ascocarps. And then our last group, or our next group, sorry, is Basidiomycota. So there's that mic again. And the way I remember these is these are your basic mushrooms, and they're named because they look like clubs. Their reproductive structures look like clubs. So your Basidiomycota or your club fungi are your basic mushrooms. So when you picture that basic kind of cap and stalk mushroom, this is what you're picturing is Basidiomycota, okay? So they have this little pedestal that is their fruiting body, and they produce basidiocarps that produce basidiospores. And so once again, same kind of processes, just different structures that are produced within that reproductive cycle. All right, some other general groupings that you can find are molds and yeast. A lot of times these fit within these other groups. And I definitely want to point out these two. Both of these are very important relationships. So we have lichens and mycorrhiza. They're both very ecologically significant. Okay, so notice that both of these are mutualistic, which means they're a win-win kind of situation. And in both cases, the fungi are providing absorption. Remember that fungi are very good at absorbing because that's how they get their nutrition. And so in the case of lichens, it's a combination of either fungi or algae or photosynthetic bacteria. Now, we actually talked about lichens back in our ecology unit, and they are ecologically significant because they are typically the pioneer species in primary succession. They can grow in very harsh conditions. And so these fungi kind of provide an environment for these unicellular photosynthetic organisms. And so the algae, because they're photosynthetic, they're providing food, and then the fungus is providing environment and absorption for the algae. So together they can create, I'll show you a picture here in a second, but they create these almost lettucey, mossy looking structures that can grow on bare rock. And then those can help create soil, and then that can lead to that progression in succession. So lichens are pretty ecologically significant because of succession. Now, mycorrhiza, okay, here we have two prefixes that I mentioned. We have myco, which is fungi. We have rhiz or rhiza, which is root. And so these are a relationship between fungi and plant roots, okay? So your vascular plants. Once again, the fungi are very absorptive, and so by having all of these hyphae around these roots, it helps absorb more water and nutrients for the plant. And then the plant, once again, is a producer, and so that plant can produce food and share that with the fungi. And so here's pictures over here on the left. These are your lichens. You can see them growing on the rocks. And then over here, what looks like a very elaborate root system is really a combination of those roots and that mycelium. And so it provides a win-win for the fungus and the plant. So here you kind of see a picture of without mycorrhiza, and here you can see that the plants are growing much better with them. So they're definitely both ecologically significant, and you do need to be familiar with those two relationships. And so the last thing we're going to talk about is the ecological impact. Because they are saprobes, many of your fungi are decomposers. That's how they get their nutrients, which helps in those nutrient cycles. We do have parasitic fungi. There's animal parasites like a ringworm, athlete's foot, yeast infections. Any type of fungal infection is called mycosis. If you put an ES, that just means plural. So mycoses are pretty much pathogenic. fungi. And what they're going to do is, because they're decomposers, they break down tissues and then absorb nutrients for food. So they can definitely be harmful. There's many fungi that can affect plants. In fact, they can really be very detrimental to agriculture. So it's something that they have to be mindful of in farming. There's also some positive roles, like the food industry. We use them to make alcohol, those yeast, and baking bread. Fungi can be a source of antibiotics. In fact, we're going to take a look at one in our lab pictures, or your lab, and it's going to be about the discovery of the very first antibiotic. And so fungi, pretty simple, basic characteristics that you need to know, as well as ecological impact. And then now we're going to switch over and finish up our lab pictures. So we've done all the protists. Hopefully you've gotten those filled in and you've at least started your research. You should have a pretty full day to do nothing but work on this lab. And so we're going to finish up with our fungus pictures. And so this is where we are. So if you don't have your lab out, go ahead and take a second and get it out. And then today, if you have time, you can continue that research, or tomorrow when you have a work day, you can continue that research. And so this was the last protist that we finished. These was your foraminifera. And so we have 4 pictures for fungi. Okay, so the first one, and one time a student pointed out that this looked like a face, and once I saw it, I can't unsee it. So this picture here, you kind of have these long strands and then these kind of tassel looking structures. But this is a picture of Penicillium notatum. And so you've probably heard of penicillin before, and you know that that is very significant to medicine. It was an accidental discovery, but man, was it a good one, because that revolutionized dealing with bacterial infections. And so I need to blow this up a little bit. Let's see. Let's zoom. All right, that's a little better. All right, so here you can see the face. There's the eyes and the nose. But you can see these long, like filamentous hyphae with the tassels. So this is what your Penicillium notatum looks like. And of course, you can do your research about that accidental discovery. All right, the next picture. Okay, now this kind of actually looks like bacteria. In fact, if this were bacteria, these would be some gram-positive cocci. But this really, really dark stained is a picture of budding yeast. And I tried to find images that look like the ones that we had for our microscopes. So I used to actually have students looking in the microscopes at these actual slides. And so rather than giving you the big blown up version like you saw in the notes, this is a picture of what you would see if we were looking at yeast under the microscope. And you've probably seen yeast in person if you've ever used it in the kitchen or for something like that, or your mom's used it. It just kind of looks like a grainy, sandy type. But if you were to blow those up under the microscope, your yeast are unicellular, and they kind of just look very, very small like that. All right, this next one, these are your zygosporangia, so your zygospores. Remember, they have these resistant structures that produce the spores. And so this is kind of what these reproductive structures of like your molds would look like. Pretty definite, easy to identify type of picture. And then the very last picture that you have is of a Basidiomycota, which, remember, those are your basic mushrooms. And if you were to take the mushroom, you have the cap and stalk. What you're looking at down here is kind of a picture of a cross section of that cap. And so you know how on the underside, if you eat mushrooms, if you pop off the top, there's gills on the underside. We're kind of looking at that microscopically. Okay, so this is just kind of a blown up version. You don't have to magnify this very much. In fact, this is probably just on low under a microscope. But you can see where that central stalk is, and then this is just a cross section of that cap. Okay, so the fungi pictures are really pretty easy to identify. And just to remind you that when you take your lab quiz, you're going to see a picture, and with that picture will be a question. It could be, what is it? or it could be one of the questions that you're having to research. It could be, what group is this in? All right, so for that lab, you really need to do a thorough job of drawing your pictures and filling out the information. You do need to complete the lab. It will be turned in the day of the lab quiz as a daily grade, to kind of hopefully help you out on those lab grades. But these pictures are going to be important because you need to recognize, first of all, what it is, and then you should be able to answer questions about it. You will also have matching sections over this information in your lab. So that's all I have for you today. That wraps up our unit, really. All you have left is to work on the lab. I would encourage you to work on the study guide. There's some great practice questions in there. And then you can also be working on that protist essay, okay? So the protist essay is due the day that we take the test, and the prokaryote, protist, and fungi lab is going to be due the day that you take the lab quiz. And so that's all I have for you guys today. I hope you have a great day, and I will see you soon.